So today, now we are going to discuss about some of the external structures and internal structures of life histories of in xylotum and other pteridophytic members. So the last class, we completed entire the external structures of that is of pteridophytes. So and also, so what is the most important points and everything is last class we are completed. So now we are going to discuss. Before going that one, already I told in what is the sporophytic evolutions in pteridophytic groups. Yeah. What type of classifications and origin of pteridophytes. Okay. So next to life cycles of in pteridophytes. Okay. All these things completed. So now we are going to discuss some members of in pteridophytes. So the members of pteridophytes, according to our syllabus, that is of in xylotum, and next one, just to wait, I'm seeing that one. Xylotum and Lycopodium and Selaginella and Equistems. So these are all the members in pteridophytic groups in our syllabus. So now we are going to discuss first one, that is of in xylotum plant. Okay. Here now you see, just I'm showing the first. This is the xylotum plant. Okay. Yes. So before going that one, you see here, this is the classification of in xylotum plant. The division of in Xylophyta, class that is of in Xylotapsida, order that is of in Xylotalis, family Xylotaceae, the genus that is in xylotum plant. This is the classifications. But here, the only most important two species is there. The xylotum is well represented only two common species, that is of in Xylotum adam, Xylotum that is of in Basidium. Two are main species is there. Yes, so now we are going to the external morphology entire the plant. So before going to discuss, here now you see this plant body. First of all, this plant body, it is in sporophytic plant body. This is the first point. Okay, from here to where, when you are going to internal external morphology in all the plants, that is in sporophytic plant body. Yes, first point. So here the sporophytic plant body means it is divided in three categories. One, it is in root system, stem system, and leaf system. This is three parts is most important point here. Yes, here now you see, but in this plant, so here this plant body, it is divided into rootless, means here we are looking the roots but not in true roots, rootless brown rhizome and aerial green shoots is there. Aerial green shoots is there. But here near about this plant, it is in 20 centimeters, so sometimes are like more than 25 or 30 centimeters also we are looking in that one. This plant body divided in three categories. One, it is in a root system, stem system, and leaves also we are looking in this one. But now we are going to clearly discuss about. So here actually rootless, this plant are in the rootless. In spite of there we are looking some hair-like structures is there, hair-like structures. So we call that is in rhizomes, rhizome. So these rhizomes are cylindrical structures is there, dichotomous branched and covered by hair-like structures. Okay. So, but here we are looking entire brown like rhizoids colors is there. And here one more important point in this one. Some of the fungi, some of the fungi associated with these roots, these rhizoids. Some of the fungi associated. That's why here the mycorrhizal fungi enters into the cortex through the rhizoids, this helping into nutritions. Some of the dichotomous branches of rhizomes grow out of the soil and develop into erect aerial green shoots. Hence, both rhizomes, aerial stems are continuous and are morphologically identical. Here, this is a, so when you are going to clear studying of an external morphology, the first rootless plant, any spite of. So here we are looking cylindrical rhizome structures is there. Okay, small hair-like structures is there. So properly here we are not looking the root system in this one. Here you are not looking to the root systems. But so here this of in above the root systems. So here you are looking one special structures is there. We call that is in rhizome. Rhizome, it is a stem modifications is there. So here this is in dichotomous branched. So now you see here this is of in dichotomous branch. Dichotomous branching means always these plants are growing two sections, going to the only two section. That's why here it is in. So we are looking that is of in dichotomous branchings. Okay. So some of the dichotomous branches of rhizome grow out of the soil and developed into the erect. Okay. So here these rhizomes developed above the soil. That's why. So here we are looking it is in some erect branched is there. Erect branched. So this is the structures is there. Yeah. Next now you see here one more. The plant body of xylotum is a sporophytic branched rhizome system and dichotomously branched cylinder upright green aerial system that bears small appendages in. So we call that is in, here we are looking this one. This is of in synangia. Means here it is in sporangia like structures. Synangia means sporangia, it is a reproductive organs, but the xylotum we are looking one special type of the structures we call that is in synangia. Synangia refers to a type of sporangium. This is also one type of sporangiums. Some fens are the relatives where the multiple sporangias are fused together into single structures. Xylotum is a genus of primitive whisk fern-like plants that belong to the family of Xylotaceae. Okay, here now you see we are looking one special type of the structures is there. So we call this is of in synangia. Synangia nothing but one type of sporangia like structures. Okay, yes. Yeah, next now you see we are going to detailed description of an aerial stem. Any one of the rhizome tips may turn upward and undergo several dichotomous to give rise to green aerial shoot. The aerial shoot or cylinder, general erect, but may be pendant in epiphytes. So now you see here this one, this is often perennial and become showy dichotomous and sometimes attain in height up to 1 meter. Generally, this plant always it goes to prostate-like systems. So sometimes it is in the, sometimes it is goes to above the soil, erect branches is there. Okay. So this is the external morphology of a xylotum plant. Yes, here now you see this of all the external morphology once again. The aerial stem it is of a multi-angular and ribbed in lower part. Xylotum nudum, the terminal part of the trace while flattened. Okay, so the sporophyll differ leaves their antigen. The fertile appendages receive the vascular bundles up to the base of sporangia. So sporangia means here we are saying that is of in synangia. This is all to the external morphology. So once again, listen here. Xylotum, it is a sporophytic plant body. Differentiated plant sporophytic means root, stem, leaf. But xylotum, it is rootless. Instead of root, here we are looking some hair-like structures is there. Above the hair-like structures, there we are looking one erect branches is there. So we call that is a rhizome. It is a cylindrical. The development it is in dichotomous branches. So here the above the dichotomous branching, there we are looking reproductive organs. We call that is of synangia. Group of sporangias is there, that is of synangias. Okay. Next, now you see we are going to the internal structures. Okay. Yes, internal structure first that is of rhizome. Rhizome means stem. The stem, aerial stem is covered by the epidermis, followed by the extensive, that is of in cortical areas, single layered endodermis, and stele. Okay, the stele is of in siphonostylic, the basal part of which becomes actinostylic in the younger branches. Here now you see this one. This is the structure of in this one. Here it is. This is in epidermis. Okay. So here these are all below the epidermis, above the stele, that is in cortex, and a center part of the section that is in stele. Okay. Yes. So the epidermis, that is entire in outer layer, single layered. This is in which outer cell walls are heavily cutinized and covered by a layer of cuticle. Cuticle is there. Here you see now one above the epidermis, one layer like structure is there, that is of cuticles. Okay. Yeah. Next one that is of cortex. You see now here this is total cortex. The cortex here we are looking two regions is there. You see one outer cortex and a middle cortex and inner cortex. Okay, two or three layers like structures is there. Just below the epidermis, that is in outer cortex. After next one that is of in middle cortex, and next one that is in inner cortex. So here I am here. The cortex is differentiated into three distinct zones. Outer zone consists of 3 to 5 rows, that is of chlorenchyma cells. Okay, with abundant spaces also we are looking that one. So middle layer that is of 4 to 5 layer, that is of sclerenchyma. Okay, so here now this is it. In middle layers is there, but it is framed by the sclerenchyma. It gives to as a mechanical support strength of in this structures. Okay. So next now you see here that is of inner cortex is there. Okay. Third one that is in inner cortex. That is inner cord. Yes. The inner zone consists of thin-walled parenchyma cells without intracellular spaces. They contains starch, some of all type of food materials also we are looking in this one. So here, this is total in cortex. So once again, the cortex, it is a three regions is there. One, it is in outer cortex, and inner middle cortex, and third one, that is in inner cortex. The first one, outer cortex. So outer cortex here now you see from three or four layers is there. So this is of in outer cortex. Okay. So this uh, outer cortex. You see now here some little intracellular spaces is there. Okay. Here we are looking intracellular spaces. And now you see here this one. This is of in middle cortex. So middle cortex, it is totally sclerenchyma cells is there. And last one, that is of inner cortex, there is no intracellular spaces also here you're looking in this one. Yeah. So next, last one, this of in stele. Stele is most important. The stele that is of in protostelic. Protostelic means that is of in without pith. Okay. So without pith, that is of in protostelic. Before going that one, this stele, it is in advanced stele. Okay. So is in protostelic, okay, without pith. The upper region, it is of in siphonostelic with pith and center. Okay, two type of the general we are saying that is of in protostelic and siphonostelic. Okay. Here, you see here this one. This is of steel. Actinosteel. You are saying here it is enough in actinosteel is this. Yeah. So yes, the pericycle is not well developed. The phloem consists of group of thin walled. Okay. So this is of in center section of the stem section cuttings. Yeah. So next one, now you see this of in this in it is of in synangium. Synangium means this of in sporophytic development. Here, this is the plant. This plant. Okay. Yes. So one is a special type of reproductive organs is there, that is of synangia. The maturity, many of the dichotomously branched aerial sporangium. The synangia located at the tip of very short axis, measuring 1 to 2 millimeters diameter and closely associated with a forked foliar appendages. At maturity, the synangia exhibits of that is of laculocidal dehiscency. So this is of in sporangiums. So here I am sharing now you see. Now you see this is of in sporangiums. Okay. So three the synangium is extremely trilobed, which internally divide into three chambers, which are interpreted as a tall sporangia, which are fused together based on their long pedicels. Okay, now you see this one, spores. Now you see when you are looking in one synangia is covered by 3 or 5 layered jacket-like structures is there. Now you see this is a one jacket-like structure is there. So after the development, the spores are comes to outside in this sporangium chambers. So this is the reproductive systems. Okay. So next now you see here. So when it comes to outside the sporangia, so here these sporangia is produced some vegetative structure. Some vegetative structure we call that is a prothallus. The prothallus of the xylotum is cylinder 1 or 2 centimeters long and 1 and 2 meters in diameter. The branch of dichotomously one or twice each branch of terminates into a single spatial cells with 4 cutting faces of pyramidal apical cells with 3 cutting faces. Okay, so now you say this is a prothallus. Okay, yes. So this prothallus, we are saying where it is in gametophyte. That's why the prothallus has no differentiation of vegetative and reproductive portions since the sex organs, both antheridia and archegonia, occur in large number of all over the gametophyte. So here this one, this is of gametophyte. Okay, so this gametophyte prothallus developed as a gametophyte. This gametophyte is terminated male and female reproductive organs. So now you say this one male, it is in antheridia. Okay, the female that is of in archegonia, male and female. So what is antheridia? So antheridia is nothing but antheridia develops from a single superficial cells. Already last class we are discussing this of in gametophyte. So this gametophyte, one of the cell, one of the section is going to modified as a male reproductive organ, that is of an antheridia. Okay. Yes. So one cell first, one of the structure is going to divide it as antheridial initial cells. Antheridial initial cells of the prothallus. The initial cell first is divided in 2 parts. Okay. So there we are looking enough in periclinal division and the superficial cells produces an outer jacket initial and an inner primary androgonial cells. So now this one, this is in antheridia. So first this structure is divided in 2 parts. So upper part and lower part. Lower part is attached to get the thallus. So get the gametophyte. And upper part it is going to periclinal anticlinal divisions and metatic divisions. So one layer is modified as a jacket layer. The below part is modified as a male reproductive organs. So we call that is often androgonial cells. Androgonial cells. So these androgonial cells are going to modified as a male reproductive, means antherozoids, male reproductive organs. Yes. So next now you see a second one, that is often archegonia. This one here it is often archegonias. Okay. So this one is of in antheridia and here it is of in archegonia. So this one, the same archegonia is also one of the structures, one of the cell. Okay, is going to modify as a archegonia cells. So here archegonia also develop from the single superficial cells of prothallus. The initial cells undergoes of periclinal divisions to form an outer primary cover cells and an inner central cells. The anticlinal divisions is followed by periclinal divisions of the outer cover cell produced to long pro. So there is a Project neck arranged 4 ventral rows of cells with 4 to 6 cells in each rows. So this is of an archegonia. Okay. The sex organ is of an antheridia, and this is of an archegonia here now. Okay. The first structures divided into ventral canal cells and other one is of neck canal cells. In between that is of an egg canal cells. So here this is of an antheridia. Okay. So this is of an anthrozoids. Okay. Androcytes is going to modify the anthrozoids. After the development, this all the spores are comes to outside these antheridial chamber, these antheridial chambers. So these spores are going to where the female reproductive organs. So when entered inside the female reproductive organs, so we call that is in fertilizations. After the fertilizations, going to modify as that is of an embryo. Okay. After fertilization, the zygote endorges are divided transversely, forming an outer epibasal cell towards the neck and inner hypobasal cells. Okay. The epibasal cells gives rise to the first rhizome, from which aerial shoots are produced. The hypobasal cell gives rise to the foot as the axis develops from the epibasal cells. The shoot apex is toward the neck. This kind of polarity in the development of embryo, that is often exoscopic, unlike the endoscopic polarity found. This structure we are looking at only, that is often lycopodiums. Okay. So here now you see this is the development diagram of. So one, it is of in foot. Okay. So that is all. Here it is in calyptra. And yes, so this is the life history of Encyclopedia. Okay, the xylotum strobules. Sporophytic plant body is produced to the strobules, means cone-like structures. Okay, so these cone-like structures are produced to which one, that is of synantia. Okay, called that is of sporangium. Sporangium mother cell. Now see here it is a meiosis. Okay, so spores, and next the spores are produced to the prothallus. Means here it is of in gametophytic stage. From here to here, this is all to the sporophytic generations. Sporophytic plant body produced to the strobules, cones, sporangium, okay, synantiums. So after the development, the spores are comes to outside in this area. Okay. Outside in this area, so these spores are germinated to new structures, new vegetative structures. We call that is of prothallus. So prothallus, after the development, because we are seeing it is in gametophytic stage. So now you see here this one, this is in gametophytic stage. Means here prothallus is modified as a gametophyte. So gametophyte, one this of in one of the cell modified as a male reproductive organ, that is in antheridia. Okay. And similarly, second part, that's the female reproductive organs. Antheridia is produced to the male reproductive organ, yeah, the sperms. When comes to outside the antheridial chambers, and next now here, egg, the archegonia. The archegonia is a formation by the egg. Okay. So both together formation, that is often fertilizations. So these fertilization, next is going to modify as a zygote. Okay. Zygote is going to modify as embryo. So this embryo is produced a new sporophytic plant body. So alternation of generations. So now here this is of sporophytic generations, and here now you see second part, that is of gametophytic generations. So this is the life history of gametophytic xylotum plant. Yes. So next, now you see we are going to one more second one, that is of in Lycopodium plant. Okay. Yes. So first, now you see external structures, external morphology of in Lycopodium plant. Yes. So first one, the Lycopodium classifications. The division that is of in Lycophyta. So next one that is of in Oligopsida, and order that is Lycopodialis, Lycopodiaceae, and so genus that is in Lycopodium plant. So the Lycopodium plant is a small, it is a cosmopolitan distributions. Okay. So near about all over the world wise, we are looking in 400 species is there. So these plant it is of in subtropical, okay, subtropical forest we are looking, or tropical and subtropical forest area we are looking these plant. So here they are commonly known as there's a club moss, okay, club moss plant. So means uh, here the leaves you are looking like moss like structures is there. That's why we are saying that is a club moss plant. Club shaped cones at the tip of the stems and branches with cones appear like a pine tree. So this is the external structures of where the occurrence of in Lycopodium plant. Yes. So now you see here I am showing the first. This is the Lycopodium plant. This is the Lycopodium plant. Yes. But sir, here these Lycopodium plant are also sporophytic plant body. Okay. The plant body it is a herbaceous and sporophyte. The most of the species of Lycopodium it is a terrestrial. So these are all the plants are growing where the damp shady places, mostly on the rocks, and also free floating hanging epiphytic plant like that is of Plagmeria. So like Plagmeria and so these Lycopodium are divided in two types of genera is there. That is of Eurostachya, and next one that is of Lepidostachya. Okay. Yes. So now you see at this one once again. So generally the species of creeping plant erect. So this is of in troubling some of the epiphytic plants. Okay. All type of the different type of the. So for example, you see when you're looking the one plant, so you're looking only one structures. For example, Azad recta indica, we're looking a long tree, or Vitex nigando, a small tree-like structures. But Lycopodium, all the Lycopodium plants are not looking the same. So one plant to other plant, lot of variation is there. That's why. So these plant, some of the epiphytic plants is there. Okay, some to the hanging plants is there. So all types of plants are looking Lycopodium plant. So when we are looking in the Lycopodium plant, based on the species, based on the structures, these Lycopodium plants are divided into categories. Now you see this of Eurostachya and next one is of Raflostachya. What is the differentiated between Eurostachya and Raflostachya? Based on the plant structures, so now you see here, the first, plants are erect, terrestrial forms, okay, and pendant in epiphytic plants. So that is of in Eurostachya, okay. So plant body is either pendant or erect. Creeping stem is absent, okay. Creeping stem is absent. Yeah, now you see a second one. They have roots only on the base of the stem. The roots always we are looking only where the base. Next one is bulbs are formed used for the vegetative reproduction system. The branching it is in dichotomous branches. So based on these structures, okay, the sporophyll are almost similar to the foliage leaves except in their size. Sporophylls. When you are looking in Eurostachya leaves, Sporophyll leaves means which contain the spores, but that type of the spores almost are similar to other foliage leaves. Okay, the strobiles are rarely found. The plants reproductive vegetatively by means that is often bulbous. So based on these structures, we are saying, sir, these are all the plants are belongs to, these lycopodium plant belongs to Eurostachya. And next one, now you see the second one. So the plant body is generally trailing or creeping with erect branches. So now here this one, plants are creeping with trailing stems. The stem profusely branched and shows of determinate growth like this. Okay? Determinate growth. The adventitious roots are arise all along the entire length of prostrate stem. The sporophyll differs from the foliage leaf. This is also most important point here. So a little bit variation, normal leaf to sporophyll leaves, size and color. Okay. So you know margins, everything is lighter, little bit differ to other normal leaves. Okay. The sporophylls organize into well-defined to the stobules borne to the long stalks. The members of this group rarely produce by to the bulbous. So this is all to the Eurostachya. Okay. Now Raphleostachya. Two type of species is there, Eurostachya and Raphleostachya. But one more important point, in all the lycopodium plants are not looking the same. One plant to other plant, little bit variation is there. Based on the plant structure, it is divided into Eurostachya and Raphleostachya. Yes. So next, now you see here. So we are going to the root systems. So first, what type of root system is there? The roots are adventitious and dichotomously branched. In the subgenus Eurostachya, adventitious roots are arise only at the base of the stem. The subgenus of in Raphleostachya, adventitious roots are present along with entire length of prostrate portions. This is the root system of in lycopodium plant. Here now you see. So next one, that is of in leaves. Okay, here the leaves are microphylls. Okay, microphylls, lanceolate with broad sessile base. They are arranged in spirals with the means. When we are looking the plant, here leaves is there. Okay, so or arrangement of two or three. Sometimes we are looking more than 6 or 7. Also we are looking in this one. But sternum, it is a hair-like structures is there. Roll-like structures and hair-like structures is there. But some of the members, it is a dimorphic leaves also we are looking. Dimorphic means two type of shapes also we are looking. The leaves are very simple, small, sessile, lanceolate with broad base. The phyllotaxy is a spiral structures is there, but some other member that is of in decussate also, roll-like structures is there. The leaves are similar, most of the species, but in some, the leaves are two sizes, that is of dimorphic, and arranged of definite vertical rows along the stem. The leaf size simple midrib, which does not reach the apex. So this is the external morphology of a lycopodium plant. Yeah. Next, now you see here, this is a one more plant is a Plagnaria. Yes, so next now you see the root systems. Now we are going to discuss about the internal structures. What type of internal structures is there? The section cutting of root. The first section cutting of roots. So the roots arise from the pericycles. Means all these roots are coming to where the pericycle is there. Pericycles, welt, and endodermis of the stem. Generally, here see epidermis is there, and next one that is often cortex is there, and here now you see this one. This often stele is there. Generally, is of three regions. One it is in epidermis, second one that is in cortex, and third one that is in stele. Okay. Yes. So first one now you see this of an outer layer that is of an epidermis. The epidermis cells are produced. Okay. Here it is in some root hair-like structures is the root hair-like structures. So in a single layer, here no any intracellular spaces also we are looking intracellular spaces. Some here it is in hair-like structures is there. In the epidermis is produced to the hair-like structures. Yeah. Yes. So next to below the epidermis above the stele. Okay, we are saying that is entire section that is in cortex. Yeah. The cortex is so many layers like structures is there. So many layers is there. Okay. One it is in outer cortex is there, and second that is of inner cortex also we are looking in this one. Okay. The outer cortex is made up of thick walled cells which gives is a mechanical support to the root systems. Okay. This is the outer cortex here. So this outer cortex is used to is a mechanical support to the root systems. And next one that is of in the inner cortex is contained that is of in parenchyma cells. Parenchyma cells. Here this one. This is all simple tissues in there. So which contain all type of that is of in food materials. These cells are total contain the food materials. Yeah. So next third port that is of steel. This is the most important steel here. You are looking in the xylem and phloem is there. And next one the steel maybe monarch. Okay. Or also that is of dioc. In most that is of dioc, the xylem it is in special one structure is there that is of C-shaped structures is there. Or sometimes there is of U-shaped structures also like. But the protoxylem at the tips of over the xylem. You see now here this one is a xylem. The phloem is present in between of U-shaped structures is there. Sometimes the root diarch on the region, but tetrarch in other regions. So this is the aerial root structures is there in Lycopodium plant. So after the root system, so now you see we are looking internal structure that is in leaf systems. So already you know very well the leaf that is of epidermis, and next one center part that is of mesophyll, and next third one that is of stele. Vascular bundles also looking in these three regions. Okay. The internal structure of leaf shows the outermost epidermis having some small hole-like structures. See that is of stomata and both surfaces. The epidermis is followed by the that is of mesophils. The mesophyll is made up of uniform all same type of parenchyma cells. The cells of the mesophyll may be angular without intracellular spaces. Small type of intraspillars also we are looking observed. The midrib has the central mass of xylem surrounded by phloems. The xylem is made up of annular of spiral tracheids. The phloem consists of parenchyma and sieve cells that are narrow endodermis, not distinct. So this is all to leaf internal structures. Yeah, next now you see we are going to the stem section cuttings. Okay, so this is also most important in section cutting. Here, this is a stem section cutting. Okay, here we are looking three regions. So one it is in epidermis, and next one that is of in cortex, and third one that is in steel. Third one that is in steel. Yes. First, now you see here the internal structure of stem. Generally, three regions is there: epidermis, cortex, and steel. Epidermis, it is a outermost layer and is one cell in thickness. Outer walls of the epidermis cells are chitinized. Stomata are present in the epidermis. Stomata are present into the epidermis. Yes. So next, now you see here. So this one, this is an epidermis. Single layer. There is no any intracellular spaces is there. And above this one, here you are looking the one wax-like structures and cuticle layer-like structures is there. Okay. Next point. Followed by the epidermis. So we are looking so cortex. Okay. Same here also we are looking in now you see first one is of an outer cortex and middle cortex and next that is of inner cortex. Three regions is this. Cortex is which varies the thickness and the structure of some structure from species to species. Okay. So the cortex is variation is there, one species, other species. So we are not looking the same type of the cortex in that one. Some the entire cortex is parenchyma, while in other the cortex divided into three concentric zones. Outer and inner portions of the cortex is made up of thick walled sclerenchyma cells. Okay, now you see this one, it's of an outer layer. Total this of sclerenchyma cells. Means after the developed cell. So here we are looking developed cells. So these developed cells is used to as a mechanical support. Okay, here we are saying that is of in sclerenchyma. Okay, yes, where the middle portion is made up of thin-walled parenchyma cells. Thin-walled parenchyma in cells. Some outer and inner cortex are made up of thin-walled cells, while the middle portion is composed of that is of in sclerified cells. Is there some special type of the cells also we are looking in this one. Some other species, the entire cortex is composed of sclerenchyma cells and is quite distinct in early stages because of Casparian thickenings on their radial walls. So maturity endodermis becomes lignified along with the cells of the inner cortex. As a result, its of identity is lost. The pericycle is beneath the endodermis. It is 3 to 4 layered and it is made up of thin walled cells. The center of the stem is occupied cylindrical vesicular tissues, which is occupied nearly half of the area in some species, in other only one part. So this one. This is the steels. Okay, center part of the portions we are saying that is of steel. So here you are looking two type of the cells is there. So one it is in xylem and phloems. Yes. Okay. This one. So generally here the arrangement of xylem is phloem varies greatly in different species. In spore links, the central core of the xylem is surrounded by the phloem. Such stele is known as protostele. This is the most important point here. So generally, entire the pteridophytic group, the stele are two types. Okay, general the stele are two types, protostele and siphonosteles. But in lycopodiums, so here you are looking that is of in protosteles. Xylem surrounded by the phloem, that is of in protosteles. Okay, so this xylem it is of an exarch. Protoxylem mosses at the periphery. The protoxylem is composed of narrow, spiral, annular, that is tracheids. Metaxylem consists of the longer scalariform tracheids. The phloem consists of sieve cells and parenchyma cells. So this is the structure of in stele. But generally, entire the lycopodium plant, the stele are four types of protostele is there. Okay. So number one, where it is in number one. See, first one observe. One it is in actinostele. Okay. Second one that is of in plactinostele, and third one that is of in mixed steels. Okay, mixed steels. So some books are given. See, one is there pectostele of polyarch actinosteles. Okay. But this one actinostele, but here it is often polyarch actinosteles. Yeah. So that is often four types of steels are also looking in this one. Four types of steels is there. So one it is of in protostele, actinostele, okay, and a polyarch actinostele, a plectosteles. Yes. So first one. So what is the protosteles? Okay. Uh, the simple condition called of actinostele. Now you see it. Actino. What is the meaning of actinosteles? This is a simple type of steel. Okay, which the central mass of xylem looks like a star, and the number of rays vary in different regions of some stem, even or in other plant. The rays of xylem are expanded towards periphery. The protoxylem is present at the tips of arms or rays, and metaxylem is arranged towards the center. In between the rays of xylem and phloem is present. So we call this stuff in actinosteles. So here now you see this one. Here I am showing the actinosteles. Okay. So yes. So these type of the steel that is of an actinostele is there. Yeah. And next one now you see second one. Okay. So here the polyarch actinostele means the lobes of the xylem are irregular or not separated into isolated stands. That is the variation of actinostele and so polyarch actinosteles. The phloem is also present in that one, but parenchyma separates it form of xylem. It form of the xylems. The the above two types of arrangement are generally found in erect and terrestrial forms. So these are in polyarch actinostele. And next now you see one more is there. That is of in plectosteles. So when we are looking in center part of the steel, here you are looking one, two, three, four. Okay. one place and other place, one, two, three, four are. So looking like that. So we call that is a plate-like structures is there. You see here. The xylem is broken into plate-like structures, which is placed more or less parallel to the surface of substratum. The xylem plates are surrounded by bands of phloem. Okay, the steel shows of alternate bonds of phloem and xylem. These are the plectosteels. Yeah. Next one, now you see here, this is the last one, that is of mixed plectosteels. Mixed means, so center part of cell, the steel you are looking from here, here, here. Okay. So all type of the place you are looking in entire the steel, that is often mixed. Xylem occurs as the stones embedded in the phloem and the whole thing looks like. So that is often steel. So all together, so all we call that is often proto steel, xylem surrounded by the phloems. So here we are looking four type of the steels is there. Okay, this is the internal structures of xylem. Yeah, yes. Okay. Next, now you see here, life history means the reproduction system of a lycopodium plant. So already we know very well the reproduction systems is often two types here. Here it is in there. Okay. So means, so sometimes you're looking some vegetative also you are looking. So the plant body produced by the asexual reproduction systems. Okay. Yes. So asexual reproduction systems, vegetative reproduction system by the gemmas or bulbous or other things there. But asexual reproduction system by the cones is there. So now you see here, this is a cones is there. Strobules means cones is there. Yeah. The lycopodium, it is a homosporic plant. The sporophytes of lycopodium are commonly known as club moss or ground pines. Okay. The produce of fruiting bodies called of sporangias. Sporangias. Okay. The sporangias is contain the spores. Sporangia is contain the spores. But the spores, it is in homospores. All type of the spores are looking the same. That's why we are saying it is in a homospores. Okay. Lycopodium, it is a homospores. The spores of one kind are produced in the sporangias. The sporophylls are fertile leaves, the deer sporangia. Okay. Where is the plant is there? Yeah, this is the plant here. Okay. So now you see here this one, this is a sporangia. Okay, spores. Here it is stobules. So one cones. So you see here all type of the sporophylls. All type of the sporophylls coming together, we call that is in cones. Okay. Group of sporophylls, that is often cones. Here group, so which contain the sporangia, we call that is sporophylls. So these sporangium is contain the spores. Okay. Spores are sporangias. Sporophylls. A group of sporophylls, that is in sporangiums. But lycopodium plant is contain same type of the spores. That's why. So we are saying that is in homospores. Yeah. So now we are going the detail about in this one. Yes. The sporophylls are fertile leaves. They bear the sporangia, and majority of species of sporophylls are organized as strobilus or that is cones at the tip of the branches or main stem. These each cones are the central axis around which numerous sporophylls are spiral arranged. Sporangias are borne in singly on the upper side of leaves near the base in the leaf axil or on the stem just above the leaf. Okay, yes. So this plant, this is the sections. This is the leaves or above the leaves, tip of the branches. Here we are looking the development of that is sporangia. That's a cones. Okay, these cones are contained sporophylls. Okay. Yes, sir. These sporophyll shows are also variation in different species. Why means in all the plants are not looking the same. That's why. That's why. So these are all the sporophylls also. We are looking one plant to other plant. Lot of variation is this. In the most of the species of subgenera, the dictionaries of Eurostachia, the sporophylls are very similar to the vegetative leaves and they are not arranged into distinct the strobules. The sporangia are exposed between the sporophylls. In some species, the sporophylls throw green or smaller size than the vegetative leaves are the fertile or sterile regions alternate on the stem and branches throughout their length. So these sporophylls of this continue into serve the photosynthetic organs after the spores are shed. Okay. So this is of in Eurostachia. So when we are looking in Rafflostachia, the subgenus of Rafflostachia sporophylls are arranged gated together forming distinct strobules. Okay. The cones are apex of the main stem of lateral branches. Okay. So sporophylls are different from vegetative leaves. So this is the variation from Rafflostachia to and Eurostachias. Yes, now you see here this one. Okay, variation to this ones. Yes, so now you see here this one, this one in sporangiums. The sporangia is located on the adaxial side of the sporophyll and near its base. So for example, this is the sporophyll leaves. In the sporophyll leaves, so here we are looking base of the sporophyll leaves, we are looking in sporangiums. Okay. Yes. So next the mature sporangia is what type of shape that is often kidney shaped, yellow or orange colored with one stalk-like structures also we are looking with the sporangias. The wall of the sporangia is several layered and the innermost layer is called of in tapetum. This one here it is in innermost layer. This of in we call tapetum means it is a just like a epidermis structure, just like it is a protection layers. Okay, so the sporangia spores are produced. So this is of in sporangias. Sir, how the development of sporangia? How the development of sporangia means here? Sir, this is the sporangia development. It is new sporangia development. The development of sporangia, this is new sporangia type. Mean the sporangia is comes to one group of surface cells. Okay. So development of sporangia, say one initiated one cell is there. Okay. Initiated when the sporophyll is formed by the embryonic cells, embryonic cells. Yes. So first that cell is divided transfer sections. So one it is sporangial initial cells. Okay. So initial cells are arrives the adaxial surface of the young sporophyll towards in the base. So but here it is going to periclinal divisions. Okay. Undergoes it is a periclinal divisions forming two layer of rows of daughter cells. Outer row of the daughter cell produce the main part of the sporangia, while the inner daughter cell are contribute to the stalk and the basal part of the sporangia, basal part of the sporangiums. Yeah, next one. So outer daughter cells are divided periclinal divisions. So that is often how much of development you are looking, that much of development you are looking in that area. After the development formation, the sporangiums. So inside the sporangiums, The formation one special type of the cells we call that is of in sporocytes. So mean that is of in sporophytes mother cells. So these spores mother cells are forming in the differentiation which one there is of nutritive tissues called of in tapetum cells is there. Tapetum cells takes place. This formed so partially from the innermost layer it is a jacket layers is there. How much of taking the food material from to the gametophyte, that much of development we are looking in this one. So after the development, this jacket layer is destroyed. So these spores are comes to outside in sporangium chambers, dehiscence of sporangiums. When the sporangium is mature, the wall of the sporangium is derives of causing strains on the wall. The wall of the sporangium is breaks transversely along in the stomium, liberating the spores. Okay, you see next. So when they destroy this cover, the spores are comes outside in this area. The spores are what type of shape is there? 0.03 to 0.05 diameter. The spores are that is of which one of tri-radiate range. The spore wall is of sculptured exhibiting various patterns. The spores represent the first cell is gametophytic generations. Yeah, when the spores are comes to outside in this area, so it is taking food material from the gametophyte from the where the environment hygroscopic method. So after the development, we call that is a gametophyte, prothallus or gametophyte. Okay, but here, so the spores are divided into horizontal to oblique planes to form a group of cells with apical cells showing the two cutting faces. The development of gametophyte behind the five cells or stage is dependent on the entry of the phycomys, some type of the fungi is there into the basal cells. After the entry of the fungus, the apical cells divide very fastly on the both forms of meristematic segments, which develops into prothallus. Complete prothallus. So we are saying that is a gametophytic stage. Okay, but here also, so all type of the lycopodium spores, lycopodium gametophyte are not looking the same. But here we are looking so different type of the gametophytes is there. So that is of in three types of gametophytes are looking in this one. Okay. Yes. So first now you see here the gametophytic stage. So here the gametophytic stage here I'm seeing one gametophytic stage is there. Now you see one gametophyte, one type of gametophyte. This one. This is one more gametophytic stage is there. Okay. Now we're going to detail about yes, the gametophyte it is of an exosporic develops outside the spores. Outside the spore, the spores are swell in size, absorbing water. The first division result in the formation of two cells, a small rhizoidal cells, and second that is of in larger cells. So in this stage, the spore swells and become globose, breaking the spores wall through the triradiate ridge. The rhizoidal cells does not divide further and soon loses its contents. The larger cells divide in two cells by the vertical wall. The cell which adjacent in the rhizoidal cells is called basal cell, and on the which is away from the rhizoidal cells is termed as apical cell. The basal cell does not divide any more. Yes, further apical cell undergoes two further divisions and form an apical cells with two cutting faces at the stage. The young prothallus consists of two five cells. So after the five cells, it is going to the lot of vegetative structures. We call that is of gametophyte. Okay. Yes. So now The gametophyte prothallus, different type of the shapes is there. The great variation from and structure of different species is there. On the basis of their structure, their intergrading types of prothallus are recognized. So types is there. One, it is in prothallus. One prothallus, it is of in heterotropic and usually annual. It grows on the surface of the soil. This is the first point. Here, the gametophyte, lot of variation is there. For example, you see here, which one there is often Lycopodium cernum, for example. The prothallus or gametophyte is short-lived and very small in diameter. The basal part of the soil and filled with endophytic mycorrhiza. Okay, so this is one type of gametophyte. Here I'm going the detail about in this one. So where the surface of the soil, the prothallus is a short cylindrical underground part. It is associated with the fungus and gives rise to the rhizoids. Okay, and next to that is of upper part of the prothallus is lobed and green. The bears Antheridia archigonia at the base of green lobes. So this is one type of the thallus. Okay, you see now the young leaves, prothallus and rhizoids. Okay, yeah. Next, second one. One more is there where we are looking at the creeping species. Okay, the creeping species of prothallus of gametophyte of Lycopodium. It is often subterranean and get buried in the soil. The prothallus is colorless and saprophytic. This one, saprophytic. The first one is of in soil surface, but this is in saprophytic. And it may be top shaped and there is often current shaped and bears the rhizoids in the lower regions. The gametophyte obtains nutrition from the endophytic fungi present in the conical part. Okay, so lycopod, for example, Lycopodium clavitum, the sex organs are present in the central part of the upper surface. In the sex organs are the lobes present at the top of cylindrical portions. So this is one type of gametophytic stage. Yeah. So next one more is there. That's often when we are looking in the epiphytic species is there. It grows saprophytic on tree trunks just below the surface of the humus. Okay, the prothallus consists of a regularly shaped tuberous body with the several serundal arms. The gametophyte is saprophytic and obtains nutrition from endophytic fungi. The sex organs develop on the upper surface of the cylindrical structures. So this is one type of gametophyte. Means here, so we are looking three types of gametophytes. All the gametophytes are not looking the same. One, it is in surface of the thallus. Okay. So second one, that is of in saprophytic, and the third one, that is of in saprophytically, but where the tree trunks. So we are looking at three types of gametophyte in Lycopodium plant. Yes. So now you see here, sir. Next point, the sex organs. This is often you see here. Now the sex organs. That is in sporophytic plant body. This is in gametophytic plant body. So gametophyte plant bodies produce the gametes. Gametes here. The gametophyte are The gametophytes are monoecious, consist of both sex organs in the same organisms. This gametophyte, this gametophyte is contain two type of the sex organs. One it is in male and females. Okay, so you see now here where this one, antheridia is there. Next to that is of an archegonia also looking in this one. Antheridia and archegonia is there. Yes, so now we are going to detail about in the antheridia. Antheridia are both sexual reproductive organs. Antheridia and archegonia are present on the same prothallus in large numbers. Antheridia develops earlier than archegonia. In species with green aerial type of prothallus, antheridia and archegonia are generally intermingled, whereas the subterranean forms of sex organs are segregated into different groups. Okay, so this is the sex organs are development. Yes, so now first we are going to discuss about in antheridia. So this is antheridia. The antheridia is a well differentiated into outer jacket, whereas this is of an antheridia. Okay, now you see here, this is in antheridia is this. The antheridia, it is a antheridia first initial in the superficial epidermal cells here. For example, here we are looking in this is a gametophytics is there. This is of an initial cell is there. So these initial cells, okay, divides transferally into outer jacket initial and inner primary androgonial cells. So these cells are divided in two regions. So one it is in jacket initial cells is there, and next one that is of inner primary androgonial cells is there. Okay, so the jacket initial divides of anticlinally forming the jacket layers. The jacket layer is of triangular opercular cells at the center. The primary androgonial cells divides and redivides, producing in the large number of androcytes. Androcytes. See, so how much of development we are looking in this one? That much of development, development we are looking in this one. You see, antheridia and the sperm mother cell develop into the antherozoids, which possesses two flagella at the narrow anterior end. The sperms are liberated by the breaking down of opercular cells. See, so now you see this one, this of antheridia. After the development, antheridia comes out in antheridia chambers. Yes, so next one now you see here, this is of an archegonia. Okay, the archegonia is embedded in prothallus and neck. Same, archegonia means it is a ventral canal cells is there, and neck canal cells is there, and covered cells also there. How the development means? Same type of the development. Okay, initial cells, the first initial cell is going to anticlinal division, periclinal divisions. Okay, yes, so the next one, the archegonia is archegonia are sunken, where which is only necks protecting. Okay, so protruding out, the archegonia is a subterranean gametophyte or cylindrical structures with a long neck, while the archegonia of surface living type or short with neck and 3 to 4 cells in length. The venter contains the venter canal cells and a egg. The neck contains 6 to 30 neck canal cells, and the long necked form the usually one in the short neck form. At the time of fertilizations, the venter canal cells and neck canal cells so disorganized so that the passage is formed The neck cells at the tip of separate. Okay, so when the anthrozoids are comes to outside antherial chambers, it goes to where the female reproductive organ. Now you said this is a Archegonia. Same. Ventral canal cells is there and neck canal cells is there and cover cell also you are looking. Archegonia is embedded in the prothallus. Now you see here. The neck and projected to outer surface. It consists of neck canal cells and neck and ventral canal cells and egg in the basal ventrals. Okay. Yes. After the fertilizations, here the formations. Okay. So fertilization takes place by the presence of water. The anthrozoids are attached to where the attached to means the mean this archegonia is produced to some type of the chemical that is of chemotatically. Okay, the anthrozoids are attached to the chemotatically because of showing the viscous fluid from the archegonia. The fluid contains of citric acid of citrates. The fertilization result in the formation of in diploid zygote. Okay, now you see this of in zygote. The zygote after the formation, the zygote develops into the embryo. The earliest divisions zygote are similar in most species, but they show variation later embryonic development. This is of an embryo. Okay. So this embryo develops. Yes, so in the subterranean non-green gametophyte bearing the species of embryo has prominent foot for absorption of nutrients from gametophyte. And the species having green gametophyte, the embryo has a small foot which is not an sufficient absorbing organ, but it has green parenchyma cells, that is often protocom and protophilus, which can synthesize food independently. Okay. After the fertilizations, formation of the zygote is produced to the new plant. You see now here this one. So this embryo develops the stem and cotyledons, the first leaf, which grows upwards from the gametophyte. The first plates of the leaves in scales. Okay, so the species with green gametophytes of we call it the protocol. This is also most important point here. The neutral structures is developed. It contains of mycorrhizal fungi. Okay, so now you see here this is the life cycle of in Lycopodium. Sir, this is a Lycopodium sporophytic plant body. Strobulus, Strobulus formation by the sporophytes, sporangia, spore mother cells, meiosis, spores, and prothallus are gametophyte. Okay, so this prothallus is produced to the antheridia, and here now you see this one, this of in archegonia produce the eggs, comes to sperms, both together fertilizations and the formation of oospores, and next where it is in embryo and young sporophytic generations. Sporophytes are here. This is the life cycle of a Lycopodium plant. From here to here, this is of in sporophytic generations. Here to here, this one, this of in gametophytic generations. So this is the life cycle of in sporophyte and xylotums. Okay. So tomorrow we'll discuss to Equisetum and Marsilia plant. Yeah. Okay. Thank you very much.